Good morning, good afternoon, and good evening. This is our fifth workshop of the year. We want to thank you all for joining us and let you know that we greatly appreciate you taking time out of your busy day to join us. I'm Tom Concolino, and I'm here with our presenters, Angela Criswell and Aya Takase. Thank you all for attending Rigaku's virtual workshop series on X-ray computed tomography. This is the second part of our session on micro-CT data collection techniques. And as with our previous session, this session will be taking place in the lab with Angela and Aya reviewing in real time how to collect micro-CT data for a variety of sample types. Now, please note, if you missed any of the previous workshops, you can view them on the Rigaku website. But before we start, a few housekeeping items. As far as today goes, this is going to be an interactive session, and we'll be taking your questions live during the webcast. So please don't wait until the end to ask. And as usual, please submit those questions via the Q&A button at the bottom of your screen. We won't be monitoring the raised hand function, and I'll be posting relevant links in the chat window. Now, we'll be trying to answer as many questions as we can during the workshop, and we'll respond to any unanswered questions after the session is complete. Now, if for whatever reason you have difficulty viewing the webinar live, please note that it is being recorded, and you'll be able to view the recording beginning tomorrow. Okay, and with that said, I'll turn it over to Angela and Aya.
Thanks, Tom. Thanks, Aya, and thank you to everyone for joining us for the presentation. So like Tom mentioned, today we're presenting live from our office in the Woodlands, where we're going to be giving a live demonstration on micro-CT data collection techniques using Rigaku's CT Lab HX system. And the things that we'll cover today are X-ray data collection for organic materials. We'll look at some sample mount materials that we use in our laboratory, and then we'll finish up by looking at an in situ micro-CT experiment, or basically a time-resolved experiment. So before we get started, I just want to recap the system that we'll be using. So in a moment, you'll see the actual system, but this shows you a bit of what's going on inside. So upstream, you have the X-ray source. It produces a cone-shaped beam, and downstream of that, there's a sample stage, and this rotates during the data collection. And there's a camera overhead so that you can see the sample. Downstream of that, we have our X-ray detector, where we'll be measuring the X-ray photons that are not absorbed by the sample, or those that are transmitted rather. So let's have a look at the system itself. So I'm going to change the share to this screen. Just confirm you see it. Yep, here it is. So this is our CT Lab HX system. And I'm just going to show you the inside of it. So I'm just going to open the door right here for a second, turn on this light, and show you some of the things where they're located that I showed you in the PowerPoint slide. So upstream here is where the X-ray source is. And you see right here where I'm pointing, that's where any filters would go. Downstream of that is the sample stage. Right now, there's not a stage that is installed, but there are a variety of options available to you, whether you're going to do a small sample or you're going to collect data for a larger sample. And then downstream of that, again, is your X-ray detector. So without further ado, I think what we'll do is we'll just come back a little bit and have a look at our first sample. Okay, so our first sample is a branch material. So it looks like this. So it's a piece of dried wood. It's organic material that was found outside of our office that we're going to image today. And to do that, because it's a larger size sample, we're going to use the larger size stage, this one, in fact. And what I've done here is I just have a cup. So I'm not using anything fancy to mount this sample. I'm just going to put it in a cup, and I'm using some paper to make sure that it's not rocking while I put it in. Okay, so that'll keep it nice and still. I'm going to put it inside the instrument. It's really easy to put the stage on. We just screw it on here. And then we're ready to go. So I'm going to take my light out. And so for our first experiment, you're going to choose the data collection conditions in this case. So that leads us into our first polling question.
Okay, so let me show you the question. So the first question we wanted to ask you is, what energy in a filter mixture is best to see details in a 2-minute scan for this organic sample? And think about what the sample is and how absorbing or not absorbing the sample is, and also think about the size of the sample to choose the energy in the filter. I'm gonna give you guys a few more seconds to think. It's looking like there is a pretty clear winner. Okay, about more than 70% of you have voted. So let me end the polling and let's take a look at the results. Okay, so most of you think 30 kV aluminum 1mm filter would be the best choice. The other choices were 30 kV no filter or 90 kV no filter, but 30 kV aluminum 1mm filter is the winner. So let's try that.
Okay, thanks so much, Aya. So it turns out that I have the filter in place already, and we're going to go to the data collection software, which looks like this. I'm just going to hide these things for now so we can have a look. So this is the control area, and this is the setup for where we set up our scans. So I already have the scan conditions set to 30 kV. I'm going to next center this sample. All right, so here's our sample. I'm just going to move this to the middle. We also want to center orthogonal to that. So I'm going to rotate to 90 degrees and move that here as well. Okay, so this is centered now, and I'm going to collect a 2-minute image just by clicking this button here. So as soon as the X-rays come on, we have an indication here above. We also have an indication if we look at our camera view. Just show that real quick. On the top of this, there's lights that come on to let us know that X-rays are on. And we can check the status of the instrument by looking at the current rotation angle. So we can keep an eye on the data collection in this case. So in talking about this sample, remember it's an organic sample and it's composed of light materials. So that means that they're best imaged with shorter energies or lower energies, like the 30 kV or 90 kV examples that we gave you in the poll. So and just to let you know, if you want to learn more about X-ray energy choice related to sample, have a look at our third webinar that I think it's called Food and Pharmaceutical Applications and presented by Aya Takase. And Tom could put a link to that for you in the chat. And I'll reference that webinar several times today. So just keep that in mind if you're interested in learning more about that. So in this case, carbon, nitrogen, oxygen, light materials, we're going to use lower energies. All right, so it's mostly finished here. And keep in mind, you're more than welcome to ask any questions throughout while we wait for it to finish.
So Angela, we did have a question come in. So obviously we don't know what this one looks like yet, or we have a pretty good idea what it looks like. This sample looked clearly dry, right? Sat on the forest floor for a while. Would you expect, a big difference if it was a fresher sample, right? That had kind of recently broken off, that, was basically maybe freshly cut off of a tree that was pruned, something like that.
Yeah, I mean, it would obviously have some water content in it that is not present right now. So there would be higher absorption perhaps, but, and not a whole lot of difference. Maybe the contrast might be challenged. And that's the hard part about organic samples, is the limited contrast. And in this case, we're going to see that in just in a second. That's our challenge. Now, it is possible to improve contrast for these types of things. And because this is no longer alive, we could have stained it with something. And of course, if something is living, you don't want to do that. It's possible that the stains themselves can be toxic. All right, so it looks like our scan is complete. And so what we're going to do is we're just going to take a quick look at the results of this scan. Okay. All right. So here is 3 2D cross sections of this material. And some of the things that we see immediately are that, we want to zoom in a little bit here. Then we can definitely see the bark area. We can see some striations inside. That would be the rings in this case. We see some cracks in this case. So it doesn't look so necessarily bad, right? And we are using a low energy, so we would expect that. So it turns out that ideally, this is not the best condition to measure. And to illustrate that, I'm just going to go back to our PowerPoint for a moment so that we can do a quick comparison of those. So I've collected scans under each of these conditions previously, and they're shown here. So here is the data collection that we did now. So this is 30 kV with aluminum, and we see similar features that we saw before. However, if we take out the aluminum filter, then we see some sharper edges here compared to, say, this crack, and perhaps in other areas. So keep in mind that the end goal of these experiments is usually segmentation and quantification in some way. So what you want to have is something that presents for clear boundaries between features inside your image, and clearly the 30 kV no filter is better. And if we look at the 90 kV, it's even better than the both of those. And so let's look at a couple of small features that illustrate that. So for example, we have a crack in the bark here. We can't see some of this feature that goes further into the bark, but we definitely see it in the 90 kV image. And again, I've already highlighted this area, that the 90 kV gives much better contrast and lower noise compared to with the 30 kV with a filter and the 30 kV alone. So 90 kV is the correct answer in this case. So I did mention that because you are challenged in contrast for organic samples, you may want to stain your sample before you measure it. And if you're interested in learning more about that, I would refer you to our webinar number 7 on life sciences. So where we looked at some staining materials and some protocols for that. And we'll also be having another webinar in December of this year where we'll look at some more sample prep. So you might come back and see us then. And let's just look at an example of a stained insect. So these are 2 different scans of a midge sample. So a midge is basically a mosquito that doesn't have the ability to bite you and suck blood. So it's missing that particular appendage. So if you just dry the sample and then image it, this is what it looks like. So we can see the muscles and these striations here throughout. We have pretty good contrast. However, the contrast for the plastic holder is about the same as for the insect itself. If instead we do something like we fix the animal and then, or the insect, and then we stain it and then dry and image it, this is what we see. So here we see a lot more detail inside, and we see the whole of this organ, which are some muscle tissues in here. We take another look at a 2D cross-section for the unstained and the stained. We see that the unstained sample is very high noise. And like I mentioned, the gray level for the sample holder is about the same for the insect, where the iodine stained sample, the contrast is just incredibly improved. So it just shows that an extra step of staining your sample can definitely help. And like I mentioned, if you are interested in doing something like this for your samples and you're looking for some guidance, feel free to reach out to Aya and I at imaging@rigaku.com with a type of sample you're interested in staining, and we can guide you to some publications. So Aya has stained materials, looking for cracks and other things, and then I've stained some organic items. So we probably have a few publications we can share. And Tom will share that web address, I'm sorry, the email address in the chat as well. All right, so that was our first sample. And now what we want to do is to go on and look at a different type of organic sample. All right, so let's go back to the screen. Going to share so you see the instrument here. And in this case, I'm just going to quickly remove this sample. We're not going to use it anymore. And I've just taped the cup on there. So you see, I can just rotate the cup. Really easy. Just some tape to adhere the sample and away you go. So our next sample is a food material. And remember, I mentioned that food and pharmaceutical webinar 3. And where Aya looked at a number of different food materials, and we'll be looking at that next. So here I have 3 packages, and one of them actually contains a candy, whereas the other ones don't. So what I want you to decide is which one of these packages contains the actual candy. Is it S, is it N, or is it R? And I is going to present a poll, so you pick, and then we'll pick, then we'll image that.
So obviously, this is not a question to test your knowledge about an X-ray CT.
There is a correct answer.
There is a correct
However,
answer. Yeah.
it is not based on anything you have learned today.
That's correct. Yeah, this one's just a fun one, and we're just looking at another soft material. Not something that's living, of course.
I'm actually wondering if we're going to have a clear winner. This should be completely split in 3 ways, right?
All right. Yeah, as it should, right?
Yeah, as it should. And this could be some sort of test for psychology.
There you go.
So we do have a winner. I mean, it is well split, but let's take a look at the results. Okay, so which one has a real candy in the wrapper? Is it S, N, or R? Looks like N is the winner.
Okay.
So let's see if we have candy in it.
All right, we're going to find out for sure. The great thing about micro CT is it's a non-destructive technology. So we can actually take a look at it without even opening it, right? So that's what we're going to do. So in this case,
you scan them all and then pick the one that has candy and eat it, right?
That's
there
what you
go.
want.
Yeah,
that's right. So I'm going to use a smaller amount here, and I'm going to use a post with a little bit of foam, and I'm just going to have some double-sided tape that I'm going to put my supposed candy with wrapper on. So I'm just going to put that in. We said in, correct?
And as in
All
Nancy,
right.
yes.
So I'm just going to put this on here and push down a little bit to make it stay and close this. So while you were taking the poll, I changed some settings. I'm also going to change the field of view. This is much smaller of course, than our branch or wood material. So we're just going to change that field of view slightly, and I'm going to turn on X-rays. So we're going to get a transmission image so that we can center this. We're going to come down a little bit. And we're going to, of course, check it out 90 degrees relative, and it's pretty well centered. Pretty much takes up the entire field of view. So we're going to go for a quick scan in this case, just quick 18 seconds, and that should give us exactly all the information we need to deduce what's inside. Okay, so just to let you know, this is a Snickers. It's like one of these little trial size. I don't know if you're familiar with that candy, but so basically it has some chocolate, it has some nougat, it has some caramel, it has some peanuts all inside. So we would expect to see several different phases for each of these materials if in fact this was the real candy. And we can tell, cheating by looking at that radiograph, that we don't see very much other than just the wrapper. And
Somebody's going to be very sad when they take a bite out of that Snicker.
That's right. So if this was something you purchased, then you would be extremely disappointed when you opened it, or your children might be, or your Halloween guests, I guess, if this is Halloween candy. So it turns out that I have replaced very carefully, one with a small piece of styrofoam, and the other one just contains air. And the sample that we see here, in fact, is completely empty. So we just see the light wrapper, not interesting at all. Well, what would it have looked like if the candy was inside? So just in case you picked one of the trick samples, I went ahead and I imaged this previously right here. Okay. So this is what the actual candy would have looked like. So you have the chocolate exterior, and you have some peanuts inside, and you have the nougat material and caramel, and you see a lot of voids. So obviously when they make this candy, they introduce a lot of air, especially in the nougat material down below. And, but we get really good contrast. This is just a quick 2-minute scan, and we could absolutely segment each of these different phases and characterize percentage-wise each of these. So we could do some quantitative things if we wanted to. All right.
Which letter was this, by the way?
This was S.
S. S
This
was
was
the right one.
S. S was the correct answer.
Okay.
So that was the right one. So one of the things I'll point out is on the bottom, we can see a little bit of this foam. It's pretty light, but that'll lead me into my next sample. So we're going to talk about some mount materials. So I've used a couple of different mounts today, a small and a large plate, and a small that just holds posts. But you can use a number of different materials to put your samples in the beam. And so I thought I'd just show you a few that we use in our laboratory. So quite often we use tape. So I use double-sided tape to adhere that candy wrapper onto the stage. We use foam a lot, mounting clay, cork material, plastic. There's paper. Sometimes things are wrapped in a paper container, and then we use a variety of waxes to mount samples. So it turns out some of these are better than others for mounting materials. And in this case, I've tried to make them all the same thickness. And basically we want to ask in this case, which one absorbs the most? So it's a very good idea for you to understand the mounting material you use and its absorption characteristics. So that leads us into our next poll.
Okay. So let me start the polling question. So the question is, which mounting material has the highest X-ray absorption? So Angela has more than 4 on her test kind of grid, but is it dental wax, a foam, mounting clay, or plastic? Which one would absorb the X-rays the most? And obviously, we're looking for something that does not absorb X-rays a lot when you're mounting a sample. But it's good to have a good idea of which one absorbs more or less.
Right.
Okay.
And the key thing is you want it to absorb less than your actual sample for sure.
Right, right. It's a relative in that
Yes.
sense too. Yeah.
Right. But always good to know when to characterize your mount materials before you use them.
Okay, I think most of you voted. So let me end the polling and share the results. Okay, so 62% of you think mounting clay is the most absorbing material.
Okay, so
I
we'll
guess
keep
we'll
an eye
find out. Yeah.
Yes. Okay, so what I'm going to do is I'm going to go back to the display here to take out my other sample. It's easy, you just unscrew, that loosens it. We can take out this, and I'm just going to put this in there, rotate it to tighten it, and it's located right inside. So for this particular example, I'm going to use a little different energy, 130 kV for this case. And I'm going to open shutter. We should have a display open up for us. And it's not quite centered, but it's pretty good. And basically what I hope you notice right away is that we don't even need to collect an image for this particular sample. We can tell immediately what the absorption characteristics are for these mounting materials. So on the top left is the clay, with below that our waxes, and on the right side is our tape and cork and other materials. But I don't expect you to remember where all of these are. So I kind of made a little cheat sheet for you. And in this case, go here, and this is basically the same thing. It just has labels on it. So all of you, or the majority of you, were correct. So clay has the highest X-ray absorption, and that's pretty much because it has some materials, like heavier materials inside of it. Whereas wax is very low absorption. It doesn't matter what kind of wax you have. In this case, I like to use dental wax. So if you've ever had braces, you'll know what that is. Here's the foam. It has very low absorption, so we use that quite a bit. Plastic, we use that quite often as well. You've seen me use it today. Tape is easy to use to adhere your sample to a stage. Cork and paper are also quite good, but you probably want to stay away from using mounting clay unless you're looking at something like rocks. Okay, so then we're going to lead into our last example for today. And to do that, I'm just going to bring you back to the instrument. Here we go. So, and what I'm going to do is show you our next sample in this case. So this, remember I mentioned at the beginning, we were going to do a bit of a time resolved. Well, this is just a simple example that you can do anywhere. I've got a pain relief pill. It's over the counter, something you can get at any sort of pharmacy or grocery store. And what we want to do today is to ask the question, what happens to it when it's ingested? Well, this particular pain reliever is marketed as fast relief. So we would expect something to happen pretty quickly whenever we put it in and start our experiment. So how are we actually going to do that? Well, what I'm going to do is I'm just going to add some water to it to simulate whenever you take the pill and it will start to dissolve. So we're going to open up. I'll just take out this sample holder, put it down up here. Oh, and by the way, I just used a piece of screen material, so screen patch, to attach all of those on there. And in this case, I'm just using an Eppendorf tube that's taped onto here and put my pill inside. And tighten. And away we go. Okay, so I still want to use 130 kV for this experiment. So I'm looking over here at this interface, but I have some smaller features in here. So I'm going to change the focus to medium, and I'm going to keep the field of view the same. And the first thing, of course, you always want to do is to center it. Now, for our next poll, we want to ask you basically, before we start this experiment, what should we be most interested in thinking about? So I is going to ask you that poll question.
Okay. So we're talking about 4D or time resolved measurements. And when you do this type of time resolved measurements, what is most important to consider? Is it use fast scan times with low energy settings? Or is it to know the time scale for sample change? Or is it to choose the field of view to view the entire sample? I think.
Got a few answers coming in.
And all of them are things to consider, but
Correct.
especially when you're doing time resolved measurements, let's think about what would be most important. Okay, I think most of you voted, so let me end the polling and let's take a look at the results. Okay, so know the time scale for sample change is most important.
And you all got exactly the correct answer. So if our time frame for the absorption of this is extremely fast, then we have to be able to collect extremely fast to see the change of the sample over time. Now, something that is very slow, where you don't need to have fast scans. Now, it is always important to choose the right energy, but you'll choose that based on the sample type. And again, I'm going to refer you to that webinar number 3 if you're interested in knowing how to do that. And then last, we asked you about the field of view. Well, you don't need the entire sample in the field of view. You just need the area of the expected change in the field of view. And depending on the resolution at which you image, that may actually be smaller than the full sample size. So the first thing we're going to do right now, before we start our time resolved experiment, is we're going to center the sample. It's always the most important thing to do, right?
And I suppose you know the time scale for the sample change in this case, right?
I do. So this is something we've done before. In that same webinar, webinar 3, I talked about 4D measurements, or these time resolved measurements as well. So you can refer to that one. I'm just going to center. It's not so important to be right in the middle because it's definitely pretty small compared to the large field of view, but we're going to look at the whole thing. So just going to move us back to 0 degrees. And turn off the x-rays. So the next thing we need to do, of course, is I need to set where I want my data to go. And then I need to start the experiment. So how do I start the experiment? So in this case, I'm just going to add a little bit of water. So pardon me inside the camera view. And I'm just going to add a small amount of water into this tube. And then I want to immediately or very quickly start the experiment. So I have set up a set of canned experiments, and I'll review them in just a second. I want to get them started. All right. So in terms of the HX software, you can set up a number of scans to collect, and that's called a job. And what you'll see, what I've done here is I've selected a job called Capsule. Basically, what this is going to do is it's going to collect 2-minute images back to back to back to back, and we're just going to observe what's happening to this pill over time. And you probably noticed I didn't collect a 0 time point. Well, that's because I did that in advance of the workshop. so that I wouldn't have to add that extra scan. So we're just going to wait and see what happens. And while we're doing that, we have plenty of time for questions in case you have some.
All right, so we did have one come in, came in a little earlier. I think it was after the first scan, and someone with an eagle eye noticed that we had a looked to be an artifact during one of the data collections. How do we account for stray radiation essentially that may have gotten picked up on the detector?
Okay, so normally there are a number of artifacts we can certainly correct for. Like for example, ring artifacts. They're usually caused by a dead pixel on the detector. And so it's very easy to get rid of them with this instrument because you just move the sample as you're collecting, and then that fills in those areas where the data is not measured. There are also some tools you can use during reconstruction to remove those. Metal artifacts, there are special corrections to deal with those during reconstruction. And as a matter of fact, many of them, that's when and where they're handled. So I think I missed that artifact. I'm not sure exactly what it was. So
I think I saw it a little bit.
Yeah.
Like especially when you do a fast scan, when you're not moving the detector to get rid of the ring artifacts, it's most pronounced at the center. And
Yes.
it could show up like a kind of noisy line in the center, vertical line. And that's usually coming from not completely corrected ring artifacts.
Right.
But as you said, if you move the detector to kind of average out multiple pixels on the detector or to unify them, then you can get rid of most of the ring artifacts.
Yeah, that's definitely an easy way to do that. So for these quick scans, we're not doing it. These are the types of scans that you would do to evaluate your initial conditions. And we're using quick scans so that we can look at more than just one sample for a workshop. For longer data collections, in fact, you're going to use what's called a step scan. And in that case, every incremental rotation step, it's going to move the sample, and then that gets rid of those artifacts. So you don't see them for the longer scans.
Okay, so while that's finishing doing its reconstruction there, we have one other question. Actually, it was asked twice. The first time was with the wood sample, and the second time was with this pill. Does sample orientation really matter? Right, laying down versus up and down versus diagonal. So if you can elaborate on that a little bit while we're still collecting other data.
Yes, absolutely. So if you've seen some of our earlier workshops, then you would have observed that absolutely for some samples, orientation is extremely important. So I would recommend you go back to see our first micro CT workshop, where we looked at that in the case of one sample. So basically, when you're thinking about how to orient the sample, one of the things that you want to make sure of is that the path length through your sample is pretty much the same throughout the entire scan. So if, for example, I put the bark material here, the length that passes through in one orientation is different than another, so there might be some scaling issues. Now, reconstruction takes care of a lot of these things, but you can make life easier on yourself by doing the best experiment possible in advance. So definitely consider the orientation of your sample whenever you're putting it in. And you might want to take a quick scan, change the orientation, and take another quick scan. That's perfectly reasonable thing to do. All right, so it looks like we have a total of 3 different time points that have been collected. And so now let's just have a quick look at them. So the first thing we want to do is to look at the 0 time point, sorry. So here is the previously collected scan for this pill. And let's just have a look at it. So first thing is, let's look at the bottom either the bottom left or the bottom right CT cross section. And so inside the pill, you see its active ingredient with some sort of filler material. And it's surrounded by a very thin coat, which you can barely see, that covers the entire pill. And then on top of that, there are caps that are placed over the ends of the pill. So you'll see one cap on the top and one cap on the bottom. And in the middle, there's a gap between them. So you can see the airspace and the gap between the cap and the rest of the pill and the pill coating. So we can look at that in 3D, for example, and that little gap is going to be a little more pronounced in this 3D image. There it is. Okay. All right. So, well, here we go. So this is our pill. I'm going to rotate it, get a little better field of view here, a little better viewpoint, and I'm going to zoom in. So the red portion, the predominantly red portion here are those end caps that are on. And then we see this gap here. Now in this gap, of course, there's a coating that's exposed. And if this were a fast acting, you might expect that's basically the target area that we're going to see the most change to begin with is in this gap here. So we can have a look at it this way where we cut it away and we can see the coatings. We can see the space here in the pill. And let's look at what happens after just 2 minutes of exposure. So we're going to go back to here. We've got quite a few already piling up to review. All right, so this is the 2-minute time point, and just going to zoom in a little bit, and we can see the water. And right around in this area, we can't see really the cap so much anymore. On the ends of the pill, everything is still pretty much intact, but we're starting to see some dissolution in here on the sides. Let's just have a look and see what 4 minutes looks like. Oh, now we're really starting to see some things happen. So everywhere in contact with the water, things seem to be coming apart. And we see some contrast difference in the center of the pill, like perhaps there's a void that's starting to fill in. So let's have a look at that and see what's happening. We do see on the ends, however, that the air gaps are still there, although at the bottom here, bottom left, you do see some of the cap dissolving. How about 6 minutes? Oh, now there's a lot of change that's happening here. So we can definitely see that there's a gap. So as we expected, we are seeing the pill dissolve basically from the middle outward. And we see the buckling of the cap material around the pill. And so it's starting to dissolve. So if you were taking this pain medication and you had a horrible headache, then 6 minutes, you'll know that you're starting to release some of the active ingredient and relieve your pain quickly. Now, one thing I'll tell you is that this is a generic brand, and there are other brand name fast-acting pills. And I chose an experiment on this, very similar to this, in that webinar that I've mentioned a couple of times. And in that case, it's very difficult to capture that with a 2-minute scan because things are already exploding out of the pill within that time frame. So this is an easier experiment to do just by the fact that it's generic and it happens a bit slower, but it's still pretty fast. Now, rather than go through all of the different time points in this way, I wanted to show you how we might look at this 4D experiment using a different program. So in this case, we'll be using a program called Dragonfly, to review more time points. Dragonfly is written by Object Research Systems, and it's a common program that we use for segmentation and quantitative analyses. If you're interested in learning more about Dragonfly, Tom will put a link to that program in the chat window, or you can have a look at our interactive Dragonfly workshop from last month that Aya gave, and Tom can include a link to that as well. So I'm just already have that particular data set series loaded, and I've created a 4D image in this case for all the time points. So what I've done is I have the first 8 minutes, 10 minutes, and then I skip to 20 minutes. And we're just going to page through those. So this should look familiar to you. So this is the initial pill that has not been exposed to any water yet. And we can see the gap here in the top left. So this is a 3D volume view. And the other views are just 2D cross sections that are orthogonal to one another. And we can see the air gap between the cap and the coated pill. If we go to time point 2 minutes, then we see that, again, like we just observed in our repeat experiment, you see the water and some dissolution that's taking place around the pill. In the top right, I want to show you, in this view, I colored it so that you could see a little bit better this striation here. And this is the dissolution of the pill that we can see a little bit better by using different coloring. How about the next time point? Well, you'll see in this case, this crack or void in the middle happens much faster. That's probably related to the amount of water or the location of the water. And you also see, again, some bulging here of the outside portion where these end caps are located. So if we wanted to, we could quantify this and look at how it falls apart. Our next image in the time scale, and I'm just paging through each of these, and there's also a slider bar that I could use to go through all the time points, and this gap is even more pronounced, and we're starting to see some what looks like cracking in other areas in the interior of the pill. We can go to the next time point, and we see now this is changing in color, and all of the interior components that are in this section are now starting to dissolve as well. And again, not much difference in change. And then finally here, we see lots of cracks and things happening throughout the pill. So we could follow this for a number of minutes and vary the amount of water that we applied and investigate that a little bit. So that's an example of a 4D experiment or time resolve experiment that's very easy to do in a laboratory. All right, so, and with that, I want to go back to our PowerPoint presentation where we want to wrap things up essentially. All right, so just to recap today's workshop, the things that we covered were some data collection for organic materials. We looked at some different sample mount materials, and we did a quick time resolve experiment with our painkiller pill. And with that, I'm going to wrap things up, but we do have some time for more questions.
Okay, so we do have time for a short Q&A. It's actually been a little bit quieter today on the question side, but I did want to reiterate just a few things. Is there were a lot of comments coming through the chat, and so I just want to make sure everybody is able to follow through with all the links and so on, and that they'll be posted tomorrow as well. We did get a question actually through the chat here. It just came in. Do you expect similar results if you fully submerge the pill, Angela, in water?
Probably not. And that might be a variable that you use, the quantity or volume of water that you use to do your experiment. In this case, I sort of canned it a little bit in that I knew how it would behave. And I knew that I could maintain some good contrast if I didn't fully submerge it. So absolutely. I mean, you want to fully submerge it to see if the end caps respond at the same rate as the middle of the pill. That's a very good point.
It actually was a different painkiller. I used the brand name one to collect the data for the third webinar Angela has been talking about. And for that experiment, I actually filled the container. It's small, but I filled it with water, so the tablet was submerged in the water. And what you see is similar, but I think if this rapid release, kind of band or opening of the coating or the casing, if that part is partially in the water, you will see kind of different reaction. But if the entire band is wet, then I think what you will see is more or less the same.
Okay, all right. So also in the chat, I'm putting a link to the quick reference sheet that we just, that was on the website when you register, but just to make sure everybody has quick access to that. And also registration for our future, our next workshop in August. So we don't have any more questions at this point. So, we will get back to you. If you do have additional questions, you can send them to imaging@rigaku.com. And that was also posted in the chat. So as I said earlier, a recording of the webinar will be available tomorrow, and an email will go out to all registrants with instructions on how to view the recorded presentation. Links to the resources mentioned today and that were included in that chat will also be shared on that landing page. So we'll be hosting another interactive session focusing on data collection techniques on high resolution CT data collection techniques on Wednesday, August the 18th at 11 a.m. Pacific, 2 p.m. Eastern. Thank you for joining us, and we will talk again in August.
Thanks very much, guys, for joining us.
Thank you. Bye-bye now.